dear students, welcome back to the chapter Biomolecules of Plus 1 Biology. Let us continue the topic enzymes. We have discussed some features of the enzymes in the last class, and now you know enzymes are biological catalysts which are increasing the speed of biological reaction. Okay, and you also know how do the enzymes act. How can the enzymes bring the rate of the reaction very high? Okay, and you also have studied the action of enzyme, forming enzyme substrate complex, enzyme product complex, and releasing the products. Okay, in the last class we have just started the factors affecting enzyme action, and today we are discussing one of the major factors affecting enzyme action as the substrate, the concentration of the substrate. Now you know that enzyme actions are not much favored in high temperatures and high pH. Okay, for every organism based on its internal body concentration and body temperature and also the habitat temperature, every organisms are having a particular optimum value for both temperature and pH. Okay, and up to the optimum pH and optimum temperature, the increase in temperature or pH can increase the enzyme action, but beyond the optimum pH or optimum temperature, the enzyme action is not favored. Okay, not increased. So you have seen the graphical representation in the last class. Okay, for example, when we consider about the pH, in our salivary amylase is an enzyme in our mouth, buccal cavity. Okay, salivary glands release this enzyme to our buccal cavity. And this enzyme's optimum pH is 6.8. Okay, whereas the enzyme, okay, the digestive enzymes in the stomach, its optimum pH is 1.8. You know, the stomach contains HCl. Okay, likewise within our body itself, the optimum pH value varies. So, for organisms, for different organisms, according to its internal body conditions and also the external habitat conditions, the optimum value may be different. And we have discussed an example for organisms which inhabit thermal springs, hot region. Okay, their optimum temperature will be high. Anyway, optimum temperature, optimum pH, what is the value? Beyond that, if there is temperature or pH increase, it is decreasing the enzyme action. So you have seen the graph like this. Okay, our body enzymes, its optimum temperature will be 37 degrees Celsius. Okay, at high temperature what happens to the enzyme? The enzyme gets denatured. Okay, if it's a low temperature, what happens to the enzyme? The enzyme is actually not destroyed, but the enzymes get inactivated at low temperature. At high temperatures, enzymes are destroyed, actually they are protein. Okay, but at low temperature they are inactivated. So, low and high temperature do not favor enzyme action, and low and high pH do not favor enzyme action for most of the enzymes. Okay, and always, for all enzyme action there is an optimum value. Okay, then let's move to the next topic, how the concentration of substrate affect the enzyme action. Okay, the graph gives a different way. Here you can see along x-axis the concentration of substrate is taken, along y-axis the velocity of the reaction or the rate of the reaction is taken along y-axis, and the concentration of substrate is taken along the x-axis. What happens when there is an increase in concentration of substrate? Suppose with the increase in concentration of the substrate, what happened to the rate of the reaction? Okay, the rate of the reaction increases with the increase in the molar concentration of substrate. Okay, then up to a limit. And this limit we can call as the maximum rate, maximum velocity we can call as Vmax. And its half velocity we can call as Vmax by 2. Okay, then at this stage, what happened? Why there is no further increase? Because at this stage, all the enzymes in the particular reaction will be used. No more enzymes are free to accept what we add. If you are again adding substrate, there are no more enzymes to bind with it, because enzymes got 100% saturation. So understand, when we discuss the concentration of substrate as a factor, Okay, up to a point where there is all enzymes saturated with the substrate. All enzymes are bonded to the substrate. Then again we add substrate means no enzymes to bind it. Okay, so this is that point. That time is the highest velocity rate of reaction. We call it as Vmax. And Vmax by 2 is the half rate. Okay, then just keep in mind there is a value given by a scientist called Km constant, and there is a correct specific Km value for every enzyme, and Km value is the concentration of the substrate required to reach Vmax by 2. Okay, so Km value can be defined as the concentration of the substrate in a reaction which is required to reach half Vmax, not the maximum velocity but half of that. Okay, this Km value, getting the Km value, we can understand the rate of the reaction or the enzyme's role in that reaction. So Km value is constant for or specific for every enzyme. Okay, so anyway understand what happened to the velocity of the reaction with concentration of the substrate. When the concentration of the substrate increases, the rate of the reaction increases up to Vmax. Okay, at Vmax, all the enzymes will be saturated. Then no more addition will favor the enzyme action because there are no free enzymes to bind with the substrate. Okay, then Vmax by 2 is the half velocity, and how much or how many number of substrates are required to reach half velocity Vmax by 2 is considered as a specific value, Km value. Okay. So I think this topic is clear, which are the factors affecting enzyme action. Temperature, pH and concentration of the substrate. Okay, you know, during enzyme action, the substrate binds to the active site of the enzyme. So the active site is specially, imagine it is specially designed for the substrate to fit. Okay, then suppose there is some other molecule similar to this substrate which has similar structural configuration like substrate. Then this enzyme site can be occupied by that other molecule too. Is it not? Just imagine, equal to the substrate, there is another molecule with the same structural configuration like the substrate. If so, that substrate also can come and bind to the enzyme. What happens in such case? There are such cases occurring in enzyme action. We can call that as inhibition, enzyme inhibition. So, in order to understand the enzyme action better, just listen to this picture representation. This is our enzyme. Suppose I have taken the example, the enzyme name is succinate dehydrogenase. Succinate dehydrogenase. So what's the name of this enzyme? This enzyme is succinate dehydrogenase enzyme. Okay, this enzyme is having a specially designed active site. Okay, so its normal substrate is succinate dehydrogenase enzyme. Its normal substrate is succinate. So the succinate will be having, okay, suppose I am just drawing like this is to correctly fit here. So what is this substance? This molecule is succinate. Okay, it has a site, okay, it has the structure which can correctly fit into the enzyme active site. Then imagine there is another molecule with the same structural configuration and the name of the molecule is malonate. Malonic acid, succinic acid. Okay, so normally this enzyme succinate dehydrogenase is catalyzing the binding of this succinate to here. But there is a presence of another molecule, malonate, which has structural similarity to this. Okay, then what happens? Instead of this succinate, it can come and bind to the enzyme. Just imagine, this is the enzyme action. This enzyme plus this succinate. But this is inhibiting that enzyme action. Okay, it binds to enzyme instead of succinate. Then what is this? This is the substrate molecule. Okay, whereas this is the inhibitor molecule. And this type of inhibition is called competitive inhibition. Okay, this type of enzyme inhibition, enzyme action inhibition is known as competitive inhibition. Tell me, what is competitive inhibition? It's a type of inhibition of enzyme action in which the inhibitor competes with the substrate for the active sites of the enzyme. Understand? Here the inhibitor competes with the substrate. This is a substrate for the active sites of the enzyme. Then who wins? If this one wins, that is an inhibition. If this one wins, that is an enzyme action. So enzyme action can be inhibited by some molecules. Okay, so who is the inhibitor here? It is malonic acid, or malonate is the inhibitor, and which is inhibiting the binding of succinate with the succinate dehydrogenase. Okay, and you know what happened when we take cyanide. Okay, cyanide intake of cyanide is leading to sudden death. It's actually blocking an enzyme action. It's an inhibitor of an enzyme action. Okay, we can just move to the next concept. The different types of enzymes. The classification of enzymes. Okay, enzymes are classified into 6 classes, 6 major classes. Again, so many subclasses are also there, and some of the subclasses you are going to learn next year. Okay, under hydrolases, nucleases, proteases. Yeah, and your digestion chapter also you have studied proteases like lipases. Okay, anyway, the major classes, 6 classes of enzymes are oxidoreductases. Here I have just starting letters I have written and is giving a card to remember. Othlil. You can remember with Othlil. O-T-H-L-I-L. Othlil. Okay. So O stands for oxidoreductases. T stands for transferases. H for hydrolases, lyases, isomerases, ligases. Then tell me, which are the 6 classes of enzymes? Enzymes are classified to 6 classes. Which are they? Oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. Then just have a very brief idea what are they. Okay, oxidoreductases are the enzymes which catalyze oxidoreduction reaction between 2 substrates. Okay, remember, oxidoreductases catalyze oxidation reduction, oxidoreduction between 2 substrates. Here just listen, in the reaction S reduced, I mean substrate 1 is in the reduced form initially, substrate 2 S dash is in the oxidized form. After the enzyme action, substrate is, first substrate is in oxidized form, second substrate in reduced form. Okay, S oxidized, S dash reduced, interchanged. Okay, oxidoreduction reaction in presence of the enzyme. So what the oxidoreductases do, it's clear I think. It catalyzes oxidation reduction between S and S dash, between 2 substrates. Initially this was reduced, but it got oxidized. The second substrate was oxidized, that got reduced. Okay. Then transferases, the second group. Here itself, name itself give you the idea. Here substrate 1 is attached with a group, substrate 2 not. Okay, and the transfer of this group to the next substrate is catalyzed by this type of enzyme. After the action of the enzyme, this G is transferred to the next substrate. So we can say transferases are the enzyme which catalyzes the transfer of a group from one substrate to other substrate. Okay, it will be clear with examples only, but this textbook is not giving any such examples, so I think it's not necessary. Okay, this way briefing is enough. Okay, then the next group, hydrolases. You are very familiar, you are going to study about the number of types in this year and next year. Hydrolases are the enzymes which catalyze hydrolytic reactions. Okay, this catalyze hydrolytic breakdown. Okay, so hydrolases are the enzymes which catalyze hydrolytic breakdown of substrates. Okay, one bond is broken and it's producing other 2 products by hydrolysis, by the removal of water. Then whereas this group, the next group, lyases, are another breakdown enzymes Okay, but they are not leading to hydrolytic breakdown. Okay, just listen its action. Here there are two groups X and Y attached. Okay, this is a substrate. Okay, with a just representation only given. Attached to two carbon, there are two side groups X and Y. And after the action of lyase, what happened? This is breaking the bond X and Y. The bonds are broken. Okay, and this is leading to double bond between carbon and carbon, and these two are released. So it's a breakdown process, but not by hydrolysis. So lyase is hydrolysis. So lyases catalyzes lysis breakdown, but process other than hydrolysis. Okay, so you can make it clear with this example. X and Y, these groups are removed by breakdown process. Okay, it leads to the double bond structure. Then isomerases, next group. Isomerases are the enzymes which is catalyzing isomerization reaction. Okay, isomerization reaction. What do you mean by? You have studied some isomers, optical isomers, geometric isomers, positional isomers. Okay, glucose and fructose are isomers, you know. Okay, I mean interconversion of one isomer to the other form. It is catalyzed by this enzyme. Okay, so isomerase enzymes catalyze the interconversion of various types of isomers like optical, geometric, positional isomers. Okay, glucose to fructose conversion or its other type of isomer conversion is catalyzed by this isomerase enzyme. So what is isomerase enzyme? Which catalyze isomerization between two isomers. Okay, then ligases. DNA ligase enzyme you are going to study later. Ligases are the enzymes which ligate two substrate, which help in the joining of two substrate to make a new bond. Okay, so ligases are the enzymes which help in making new products by joining substrates. Ligases. Okay. I think the 6 classes of enzymes are very easy to study. So learn it, and we will discuss its details in the live session. Okay, and we have one more small topic that is as the last topic of the chapter, cofactors. Just listen here. You know enzymes are mostly protein, but most of the enzymes for its action, for the catalytic action, it is associated with a non-protein part also. So the protein is okay, the protein part is there. In addition to that, most of the enzymes possess a non-protein part. That is what is called cofactor, our next topic. So what is the protein part of the enzyme called? Enzyme is chemically protein mostly, and they contain a protein part. Sure. Okay. The protein part of the enzyme is called apoenzyme. Okay. But for action, almost all enzymes require a non-protein part also. What is the non-protein part called? The non-protein part is called a cofactor. So our next topic of discussion is, what is a cofactor? Tell me, what is a cofactor? Tell me, what is a cofactor? A cofactor is the non-protein segment or non-protein component of an enzyme. Okay, the protein component is apoenzyme, non-protein component is cofactor. Then this cofactor can be of 3 types: prosthetic group, coenzyme, metal ions. Okay, there are 3 types of cofactors. The first type is called prosthetic group. What type of cofactors are called prosthetic group? Prosthetic groups are organic compounds, organic molecules. They are permanently attached to the enzyme. It is not a temporary association, it's always there with the enzyme. Okay, along with the protein part, this is permanently there. So, the prosthetic group of the enzyme is an organic compound which is permanently attached to the apoenzyme. Okay, just as an example, you can learn heme. Heme, H-A-E-M, it's an organic part. Heme is the prosthetic group of the enzyme peroxidase. So, the enzyme peroxidase. Peroxidase enzyme is the enzyme which convert H2O2, hydrogen peroxide to water and oxygen. Okay, 2H2O2, 2H2O. Okay, so just listen here. Peroxidase is an enzyme which is catalyzing the conversion of hydrogen peroxide to water and oxygen. Okay, this reaction requires this enzyme. Okay, and this enzyme will be active only with the presence of its prosthetic group heme. It's permanently there with that. It's an organic compound. Okay, heme is the prosthetic group of peroxidase enzyme. Okay, so what is the prosthetic group? Otherwise, what is a cofactor of peroxidase enzyme? The cofactor of peroxidase enzyme is a prosthetic group which is heme. It's an organic compound associated with it permanently. Only with that, if we remove that, it cannot act. Okay, so this is an example of a prosthetic group. What is the example? Heme. Not only for peroxidase enzyme, for catalase, another enzyme, catalase enzyme also, the same prosthetic group is there, heme. Okay, our next cofactor, second cofactor is coenzyme. Coenzymes are second type of cofactor. Coenzymes, example, NAD, NADP, etc. act as coenzymes. Okay, coenzyme A, coenzyme, different coenzymes are there. NAD, you are going to learn it in the physiology section. Okay, so nicotinamide adenosine dinucleotide, nicotinamide adenosine dinucleotide phosphate. Just study the example of coenzyme. NAD and NADP are two examples of coenzyme. For all these coenzymes, vitamins are having, and this type of coenzymes, vitamins are present in the structure. Vitamins are important for the structure formation. Vitamins are there. Nicotinamide means niacin is there. Coenzymes are the type of cofactors which are also organic compounds, but its association is not permanent, which is a transient association. Only at the time of enzyme action, they come and bind to the enzyme. Otherwise, the enzyme is free with only apoenzyme, but at the time of enzyme action, this NAD come and bind, or this coenzyme comes to bind with the enzyme. So listen carefully, prosthetic group and coenzyme difference. Both are, similarity, both are organic compounds. Difference, prosthetic group is a permanent association to enzyme, whereas coenzyme is temporarily associated to enzyme only at the time of catalytic action. Okay, and after that it is removed. For example, NAD, NADP, nicotinamide adenosine dinucleotide, nicotinamide adenosine dinucleotide phosphate. And our next cofactor is metal ion. Metal ions are the cofactors of many enzymes. Okay, metal ions. For example, carboxypeptidase. The enzyme role you will study later. For the enzyme carboxypeptidase, zinc is the cofactor. If zinc is removed, carboxypeptidase cannot act properly. So understand, cofactors are very important for the enzyme action, the catalytic action of enzymes. The cofactors are of three types. Okay, coenzymes, prosthetic group, both are organic compounds. Coenzymes are temporary association. After the action it is not required. Whereas prosthetic group, permanent association, and also metal ions act as the cofactors. Examples, first case, peroxidase, heme, prosthetic group. Second case, NADP, NAD, okay, as coenzymes. Okay, then third group example, metal ions, zinc is the cofactor in carboxypeptidase enzyme. Okay, and with this topic, we are just winding up the concept of enzymes. We have discussed some more things about enzymes compared to other biomolecules, but actually related to enzymes there are so many facts existing, but you learn at least these ideas as brief. Okay, so study enzymes thoroughly. Essay questions and other questions are possible from this session, and any elaboration required means we will do in the live session. Thank you.